The complement classical pathway begins when either antibody IgG or IgM find and attach themselves to their targeted antigen, creating an antigen-antibody complex. A C1 protein complex called C1QR2S2 will soon bind to the antibody portion of the antigen-antibody complex. The C1 protein complex is comprised of a C1Q subcomponent, 2 C1R subcomponents, and 2 C1S subcomponents. C1Q attaches to the antibody, while the C1R2 and C1S2 cleaves. When the C1 protein binds to the antigen-antibody complex, it becomes activated and starts the cascade of proteins that enzymatically cleaves and activates the next protein. It begins by cleaving C4 into C4A and C4B. It then cleaves C2 into C2A and C2B. C4B and C2A then bind together, creating an enzyme complex called C4B2A, also known as C3 convertase. C3 convertase cleaves the C3 protein into C3A and C3B. C3A helps in inflammation by activating mast cells, causing the release of vasoactive mediators such as histamine. The C3B portion binds to the C3 convertase, turning the C4B2A enzyme into the C4B2A3B enzyme, also known as C5 convertase. C5 convertase cleaves the C5 protein into C5A and C5B. C5A attracts macrophages and neutrophils along with activating mast cells. C5B binds with C6 and C7, allowing this new C5B6-7 complex to attach itself into the membrane of the pathogen. While in the membrane, C5b67 recruits the C8 protein, creating the C5b678 complex. The C5b678 complex then recruits and polymerizes anywhere from 12 to 22 C9 proteins, creating a pore in the membrane known as the membrane attack complex, MAC. The membrane attack complex enables water and salt ions to enter the cell, ultimately leading to osmotic lysis and the bursting of the cell.